Oh, it moved a little bit. I think. Yeah.
Oh, there you go.
Is that more just like coming out of the sediment though?
Yep.
So what can we learn by taking these rock samples?
So Josh, when you have a chance, let's put it up in front of the camera and give it a spin all the way around.
Yeah. I'll shake the dust off a little bit.
Sure. So one of the things that we will learn is the age of these rocks, which gives us insight into the geology of the area, and how this particular seamount was formed and when.
Size in the lasers there.
Great. Hold this still just 1 second. We're going to take a still shot.
Okay.
Greta, let me know when you're good.
Yes, maybe one or more.
Oh yeah, I'll get it better in the. Sorry, I'll get it better in the DSC camera for you.
Yeah, if you can put it in the middle, that would be amazing. Thanks. Beautiful.
Great.
I think we're good.
Good?
Yeah.
Okay. Yeah, if we can just keep spinning that a little bit more while we're in the main video camera, just so we can see all sides of it before we put it in the box.
So what type of rock is this?
Not being a geologist, I'm not going to get technical, but it is a piece of lava.
Igneous.
That's great. Thanks, Josh.
Okay.
Just going to go in one of the big boxes, Noel.
Yeah.
I might be able to get in one of the small ones on the starboard side, maybe.
There's the farthest aft small box is open,
Okay.
or the aft larger starboard bio box.
Okay.
We can't fit it in the small one.
All right, that's a good plan. I like that plan. Okay, I'll come wide, please.
So we have a question from one of our viewers. What do the lasers do and how do they do it?
So the lasers are basically just a really fancy ruler. They are parallel, and they're
Let me
in
know
a
when you're ready for
bracket
box
that keeps
open.
them fixed one to the other. So they are always 10 centimeters or about 4 inches apart. And wherever we point the camera, whether we zoom in or we zoom out, we always have our ruler there to know how big things are. So that's all it is. It's just a really fancy ruler.
Ah, thank you.
Yeah, we'll start. I'm going for the back little one to start, but we'll just kind of make sure ready to fit. Yeah, okay, let's start. Okay. Yeah. See if I can.
Let me double check.
Don't know if it's going to fit in there.
Yeah, they're still down.
That larger bio box
of
is
course
also
Yeah,
open.
I think it might have to be this one.
Okay.
Is there anything else in that one right now, Noel?
Not
No?
the far,
Okay.
not the aft one.
Awesome.
Okay, big one it is.
Okay, aft bio box.
Okay, we can close that. Okay. Okay. Okay, I think ready to move the ship, Jeff?
Yes.
Okay.
Fish right there.
That might be another gulper, it looks like.
That might be another gulper eel. It's gonna be, yeah, really, really hard to track.
I can see its shadow.
Oh, it's a halosaur.
Right there on the right. Is that it? Or is this somebody else? This looks like a halosaur. That's a halosaur. Yep.
Also hard
okay.
to follow.
It's not a gulper
Okay,
eel.
we'll
I think that was the same thing, but that's, yeah.
Yeah, was that
Great,
the same
thanks.
fish?
I think so.
I think
As
so,
it moved
'cause
away, it looked really
I saw
hard.
its shadow, and then it kind of came into view again.
Yeah.
Okay, okay.
I don't know. I thought Max might have manifested another gulper eel.
It's trying.
That's the dream.
Derek would be proud.
Yeah, speaking of Derek, I don't think we've seen a sea cucumber.
He's left, so now they don't come out.
Yeah.
I wish him many a sea cucumber on his days diving in Palau.
I have yet to see one. I love them.
We'll keep an eye out.
They
I
are
feel
cool.
optimistic that we will find more.
Yeah. Derek just didn't think they were very cool.
Are they less common on these kind of rocky substrates? Yeah.
They are.
Seems
yeah.
like it.
Yeah.
But maybe
The area where we were on a largely sediment-covered slope descending into the Palau Trench, there was an amazing diversity of sea cucumbers. It was fantastic. Derek didn't think so, but the rest of us did.
Well, there was very little diversity of anything else, so.
I was telling Noelle during breakfast that in high school I had a science fair project where we had to just create a poster on our favorite sea creature, and I did sea cucumbers, and it was a whole entire trifold of sea cucumbers that I cut out of cardboard and painted, and someone came up to my booth and they were like, "This looks like poop." And I was like, well, they eat it.
Yeah, they do.
This was on the east side of Palau?
Yeah.
Yeah.
So there's a story that comes from one of the states on the eastern side of Palau, and it goes that this woman was living on her own, and she didn't have any kids or a husband, so she was basically destitute, and there was a breadfruit tree that was next to her house, and she cut off a branch, and water started flowing out of it, and it was fish from the region. And the villagers saw that that was happening to her tree, so they came and cut all the branches and started harvesting or getting all the fish out of it. And eventually the flow of water flooded the village, and they went astray, and it's off the coast of New Wild. So that could have a significance to that story. That's the reason why there's nothing else but sea cucumbers, because they got sucked up by the breadfruit tree.
Wow.
Wow.
Yeah.
That's cool. Thank you for sharing.
And Matt, the coolest thing about this story, and there's actually some remains of this city right down by Niguala underwater.
Yeah. And in some cases, I mean, every civilization has a story of a flood, and that's similar to those stories in other civilizations. So.
So we have a question from for our navigator. What is the significance of moving the ship, and why do you need to call specific moves and distances?
While we're on our dive, we are obviously stopping to investigate certain targets and to collect samples, and we're panning and zooming with the cameras to inform the science party. But we also need to cover ground so that we don't stay in the same area the whole time. So we have a ship track that's been planned based on the sonar data we collected when we mapped this area yesterday. And so we are slowly moving along a pre-planned track and trying to hit spots that we think will be significant and have some interesting things to explore.
Thank you for that. And does the surface swell kind of like have an effect on how you determine these movements and these calls?
Yeah, we're keeping an eye on both the wind and the current and how those are affecting the ability of the ship to hold position and the way that the vehicles are oriented relative to the ship. So it's obviously it's a balancing act since we have vehicles that are hanging on a very small tether that is thousands of meters long. And we're trying to keep everything aligned so that we can keep our ROV nice and steady and collect really beautiful footage.
Can you zoom in, please?
Thank you.
Yeah, one of the kind of interesting things about a two-body system, ROV system, that we run here is that the sled ROV that Rye is operating is essentially its position is dictated by the ship position. And then we're tethered together, Hercules and Atalanta are tethered together, so ultimately the ship is sort of really positioning the vehicles in a precise way. And so that's why I have to be so deliberate about the movements of the ship, because that ultimately dictates where Atalanta sits, and then in turn ultimately dictates where the range that Hercules can move around in. Okay, we'll come wide, please.
Thank you for that.
Yeah. Can we actually zoom on that shrimp right there? That might just be pneumatic arsinus, but it looks, it may just be shadow under its body. I just want to see if it's the same thing. Okay, we'll zoom in, please. It is. Okay, thanks. Okay, come wide. Zoom in, please. That's awful shaggy on the end of this whip coral. Looks like it's died back and been colonized by hydroids or something.
Yeah.
Would that like eventually
break off and then the coral would still continue to be alright, or is it just going to continue to take over?
Yeah, my guess is it would eventually, without the coral maintaining the skeleton, eventually those proteinaceous little nodes in between would decay or get weak, and then it would break off and fall.
Yeah. Okay, come light, please.
So we just got a shout out in the chat from El Hideos. She was one of our science communication fellows on the NA167 expedition. Ali El, thank you so much for tuning in. She said we're all doing great and go team.
Thanks, El.
Yeah.
Hi, Ali.
Let's just zoom on that hemicorallium or paramurcied or something. I can't quite tell from this distance what that is.
And we'll zoom in, please.
Did we get any more zoom, or is that it?
There we go.
Hemicorallium. Okay, let's keep going. Thanks.
Okay, come light, please.
So it's right when I get the focal point. We go wide.
Yeah, just was settling in.
Just gotta keep you on your toes.
Zoom in one tick closer on this guy. Awesome, yeah, thank you.
So we got a question from one of our viewers. What is everyone's dream thing to see on a dive?
A whale.
A whale? Any whale? Any specific kind of whale?
Any whale.
Yeah.
Just any whale will do. Any whale will do.
I have a couple fish I really want to see. I want to see another one of the batfish like we saw over on the east side.
Wow.
That was cool.
That was really amazing.
What's a batfish? What do they look like?
It's
looks like a chicken.
It looks like a chicken. Its fins on the side, the pectoral fins, look like little feet, and they literally hop around on the ground on the bottom.
I need to look that up.
They're kind of flattened and in top to bottom, and they're just fascinating little fishes.
Oh wow, they do look like a chicken.
Like a plucked chicken.
So I would love to see a batfish. And the other thing, we haven't seen any sharks or rays yet here in Palau, or a ghost shark or something like that. And I would love to see one. That'd be great.
What do you find fascinating about ghost sharks?
Ghost sharks are really weird animals. They're ancestors of the regular sharks and rays. They have a cartilaginous skeleton like sharks and rays. But they have this really bizarre, almost beak-like mouth that's kind of under their snout. And they have these giant pectoral fins on their side that look like wings coming out, and a sharp, skinny-like tail. And they often eat all kinds of crazy inverts off the seafloor. A lot of other fishes either eat plankton or they eat, sort of shrimp and other fish and things like that. And these ghost sharks may eat all kinds of other animals as well. They may browse on maybe not corals, but all kinds of other invertebrates living on the seafloor, little worms and things like that. So they're kind of neat creatures, and they're really ancestral. They kind of go back to the first lineages of fishes on Earth.
Wow, that's awesome. Thank you for sharing. And you mentioned you wanted to see a whale. What do you like about whales?
Oh, I don't know. I grew up on Vancouver Island, and there's a lot of whales around there. And I grew up on the water, so whales have always been one of my favorite things to encounter. And a few years ago, the Nautilus encountered a juvenile sperm whale as it was diving, and I've seen that video, and I think I would probably cry.
What a special moment.
Yeah.
Wow, thank you.
And we'll zoom in, please.
I'd like to see a Nautilus. Yeah.
I would like to see that too.
These animals are endemic to Palau, and it's a living fossil. I remember as a kid, my dad would put some baits. I know that this is not a good practice now because we know better, but we would put baits in a basket and would let it go down to the deep, and then we'd basically kind of bait them to come to the shallow, and that's how we were able to see them as kids.
Wow.
But eventually they always die because they're not used to the pressure changes.
Come wide for a sec, please.
Yeah. But I would love to see one in its environment, its home, in its habitat.
Yeah. Thank you.
Can we get once you get settled, Josh, I know the current seems like it's ripping down here, but it'd be great to get a better zoom on this coral.
Yeah, I'm going to try my best here.
Awesome, thanks.
I like to see a vampire squid.
Yeah.
Oh, yeah,
wow.
They're great.
We were teased with the slightest glimpse of potentially a big fin squid last expedition.
Don't lie.
And that would be pretty cool.
It's a nice view on the still cam.
Yeah, it's cool on the still cam.
Big fin squid.
Tell us when you're good, Greta.
Okay, we can zoom in, please. And
I found out recently that there was a second one sighted on the Atlantic also.
Might just want to stop the ship for a sec.
We are not sure what this is.
Adorable.
Oh.
We do not know what kind of coral this is.
Sweet.
The little shrimp look like some sort of a thorid, lebius, or something like that. There's also some smaller pink things. They might be amphipods in the branches, and lots of brittle stars living on this coral. Those are all the little white arms. But I do not know
They're so cute.
what this coral is. I think we should, given that no one on the science chat seems to know what it is either, we should probably take a very small snip of it.
Okay. Can you back the ship up just a bit?
Yeah, I'm directly above you right now.
Yeah.
Okay.
Greta, would you say these are similar, but like different, but similar to the soft coral that we see at Soft Coral Arch?
Oh,
The ones on soft coral arch are a bit of a different shape. They look more like broccoli.
Yeah.
But these remind me more about the gorgonians, which you can usually find in the tidal channels around Palau, together with the whip corals. They really love strong current areas, and sometimes we find some of them that are like, I don't know, 3 meters.
Yeah.
And they're incredibly beautiful.
Okay.
Just because Herc and Atalanta are like right on top of each other, and we like to have Atalanta just a little bit step back. 10 meters would be perfect, yeah. It'll take a bit to swing over, but.
Could we also potentially get a zoomed out shot with the lasers to get a scale size of the whole colony?
Yeah.
Yeah, let's go wide, please.
That is large.
So pretty.
Can we get both of the lasers on the coral in the front? I don't know if we can just tilt the camera down just a little bit. There we go. Great. Okay. Yeah, this is like 70, 80 centimeters across. Okay. Josh, when you're ready, can we just get a piece off of perhaps the right-hand side?
Yep.
Is this going to be a snip and slurp?
No. Don't think so.
You know, one thing that might also help is if we could get a shot in at the base of the coral before we go and collect. I don't know if that's going to be helpful. Many of the characters are the polyps themselves, but...
Okay, yeah, we can zoom in right there, I think.
But if we just look at this attachment point. Okay, great, thanks.
Okay, go wide.
So, Jeff, what has made you determined to take the sample?
Well, in this case, nobody knows what it is. So it is very likely new.
Wow, that's exciting.
Yeah.
Menzel, how does it feel to see something like this?
I'm going to name it after myself.
It's bright. It's beautiful.
Yeah.
It's you.
It's like looking in a mirror.
So graceful. Do you also have tiny shrimp like hanging on to you sometimes?
All the time. Can't get rid of them.
Okay, let's zoom in a bit, please.
One of our viewers says it's an invasive species of English broom.
English broom.
Those land flowers, they're everywhere.
Yeah, the bright yellow.
Yeah.
Do you have that in Palau as well?
Yes, we do.
Okay, we have it very bad on Vancouver Island.
Oh,
a lot of grasshoppers love it.
Yeah. It's taking over.
I weeded a lot of that when I was a kid.
Cool. So a question from our viewers, how do corals attach to rocks? Is it like glue?
Zoom in just a little bit more, please. That's great.
Yeah, there's a... Do you want to take this, Greta?
I can talk about shallow water corals and how they attach to rocks. I'm not sure how
to... No, go for it.
Okay, so usually corals, when they born, they're like just tiny little planktonic larvae, so like tiny little corals that just float in the water. And while they float around, they can kind of sense chemical cues. They sense other algae or other organisms that attract them. So when they sense those organisms, they eventually just fall onto the rock, onto the seabed, and just metamorphose into something different. So before they were very, very simple, like larvae-like animals, and when they settle, they start to actually become corals. So the larvae will attach to the substrate, just move around a tiny bit still to find exactly the perfect spot, which usually is inside a crevice where they are protected by predation. And once they find that spot, they metamorphose into the actual coral. And so they start to divide their cells and make different kind of cells so that they can actually attach to the substrate and start growing from there.
Can we get a zoom on that before we put it in the box, just really quickly?
Are we full wide?
We are.
Okay, thanks.
And I would assume in the deep sea it would work in a similar way. They would just have different chemical cues that they will be attracted to because they have very different ways of living down here. They're very different corals than the reef-building corals in the shallow waters.
Awesome, thank you so much. I think it's such a blessing that we're able to witness something that some of our scientists have never seen or not, or don't know much about.
Great.
It's beautiful.
Okay, thank you for the zoom.
Yep.
Josh, I think the small,
wide, please.
Most aft starboard bio box.
Oh boy. Yep, I knew you were going to say that.
Yep. It's the only one that doesn't have something in it.
We might want to isolate that.
Vert thrusters.
Actually, we'll see how it goes.
Okay.
Yeah, it's the most challenging one to get to, especially with such a delicate piece.
Just let me know when you're ready for the box.
Okay. Can you isolate the laterals?
Yep.
It's on a little bit. I just don't want it on at all.
It's just one relay, right?
It's just one relay up.
Okay, laterals are isolated.
Okay, thanks. All right, we can open the box.
Okay. Starboard box coming out. I'm just gonna go slow here to see if anything.
Yeah.
Okay.
There's a big debate here in the chat.
Okay, and close it. All right, that went as good as I could have hoped. So Jeff, do you mind if I spend just a minute kind of getting the DSE kind of like a little better setup for the still shot?
Sure.
I love having like the background, and it's the kind of perch nice. I can just swing around. I think I can get a pretty cool little setup there. But yeah, that was in the back small box.
You're lined up with the porch on HD.
Okay, cool.
And Nav, can we get a waypoint for that collection? It's 008.
Copy, entering that now.
Oh, you probably don't have verts still.
Oh,
you want those?
the laterals? Yeah, let's throw those back on.
Or the laterals, sorry.
Oh, that's easier.
There you go.
That is much easier.
Easier to drive with all the thrusters.
like a little lower, but that's what the best you're gonna get.
That's nice.
Yeah, I really like these coral cutters because you can, if you just really take your time and be delicate, you can just, you know, really only touch that one little edge, and the rest of the coral just remains the same. It's pretty cool.
Yeah, that was very nicely done.
Is it keeping itself up, or is the current coming from this side?
The current seems to be kind of like swirling a little bit in the current there, it seems like. We're on a little bit of like a ridge, so maybe the current's sort of coming from a couple different directions as it swirls around.
Yeah.
Yeah.
All right, oh, a little bit more?
1 second.
Yeah, no worries.
Getting a last focus.
Take your time, yeah, let's get some good stills.
Very hard to focus today for some reason. So we need to take some more time.
Yeah, absolutely.
Oh, that's pretty good there.
So cool.
Huh?
We have a good one in focus while the sampling was going on.
Mm-hmm.
It's not as close, the close up, but it's still. Let's try here.
Might be able to change angles a little bit and get it closer to the camera.
Yeah, that might help.
Okay, I just will swing around to the right a little bit. You ready for that?
So a question from one of our viewers. What is the sort of process to determine whether or not this could be like a new species?
So one of the things you do when you're identifying a new species, first of all, you need an actual specimen. You cannot do it from imagery alone. But what you would do is you would take the specimen and you would identify all the characteristics about it. So in the case of a coral, and again, you're talking to a fish guy, but I have friends who are coral people, and you would look at the arrangement of the polyps and the spines or other structures that support those polyps on the skeleton, and a variety of other characters, and you would compare that to all the known species. And oftentimes what would happen is you would figure out what general group the coral was in, and then you'd try to use the existing keys that have all these characters in them. Say, if it has this arrangement of its skeleton, it's probably this, and if it has this other arrangement, then it's probably this other thing. But perhaps you find this specimen and it doesn't match either one. In other words, it has a combination of different traits, different characters that just doesn't align with all the species that we already know about, and that suggests that it's new.
Let me try and, why don't I try and swing around and see if I can get a better...
Of course, that's for the morphology, and then in addition,
Max,
we'll
the
be
viewers are
preserving
asking
these
if they could,
animals
if
and
we could
alcohol.
zoom in the main camera while we're sitting
and
here.
we would use genetics.
If not, it's okay, but they're just asking.
And we would look at molecular markers, molecular
Yeah,
characters to
looks
see
like
if
we're
in fact this
headed out now, but
specimen was a species that was new.
I'm going to try and just...
That does get challenging at times because sometimes the specimens that we have in museums were collected before the genetic approaches really took off, and so there aren't any molecular data for a given species. So that makes that a little harder sometimes.
Okay.
Do you know how long it could take to determine if it's a new species?
Them, so yeah.
Well, it'll probably be a little while before the specimens make it back to the museum, and it takes some time for them to be ascensioned and to be archived, so that they all have specimen numbers and that sort of thing. And then they can be loaned out to a taxonomist. A taxonomist might have to spend months looking at this specimen and others before they were able to write up their results and publish something. So the long and short of it is it probably is going to take a couple of years before any kind of actual formal description of a new species would come out, if it is in fact a new species. We don't know.
Hey, Greta, can you give me the preview back, and I'll see if I can get a little closer, the camera a little closer. Oh, that did not work at all. A little higher on the side. The camera's propped up a little higher. Maybe this side's not going to work. No. Go back over here.
There you go.
Yep, there's another cut-through eel.
Oh, nice picture.
Oh, that is nice. The coral and the eel in the background.
I can't get it quite down into frame and stay like real close to it because it's up a little higher. Yeah. Well, I think what we got before is probably the best spot because I can't quite get it down into frame and be nice and close to it because of the orientation of how it's sitting with the rocks around it. But I could definitely do some more HD zoom if you want, or we can move on, whichever.
Yeah, I think we need to move on now.
Okay, roger.
So we have a question for Greta. What is the purpose of the still camera?
Okay, we can move the ship. Yep.
So the purpose of the still camera is to take high resolution pictures of particularly interesting specimens that we find out here. And we usually take pictures of interesting sightings that we see, but also to the sampling. So every time sampling of something happens, we need to take still pictures of that so that we can track what we are doing.
Awesome. And will those images be available publicly?
There's usually a large number of them. I don't know where those end up. Maybe Noel knows. Do the still images from the cameras end up online or publicly available somewhere?
It depends. A lot of them do end up being featured photos on our website and promoted with the expedition. So keep an eye out for some imagery on the web pages, as well as widely used in things like our annual reports and other types of publications like that.
Reminds me of like having a flower in your hair.
Wondering if the coral is Norella or something like that. I think it's a sponge is another Regadrella. Oh, maybe not though, because it's covered on the top. I'm not sure.
Yo, come wide, please.
Okay. Old attachment point of sponges. Yep, there's a crinoid down there.
A cliff?
This is just a ridge of rock coming down. I can see Talus Field on the other side there.
You can almost imagine what it looked like when it was forming.
It's interesting to see that the bathymetry doesn't pick up these small features.
Right. Yeah, the pixel size on the bathymetry from the multi-beam is quite large, and so yeah, all of these kinds of things get missed. Let's try to get a zoom on all these little white things on the side of the rocks there.
Sure.
Maybe they're all just old attachment points. There's a tremendous number of them.
Zoom in, please.
I wonder what happened to the organisms hanging from there.
Yeah, that's what it looks like. It looks like these were just past points of attachment for corals or sponges, and they have fallen off the wall. Okay, we can zoom out.
Yeah, come on.
Is there a way to know how old those are?
If you can date coral material, and a number of these deep water corals the colonies can be well over 1,000 years old. I don't think there is nearly as much chemistry available for the sponges to be able to date the sponges, but there's some suggestion that they could be quite old as well.
Okay.
What is that thing down there?
Oh, there's your cucumber.
Another one.
Sir.
Oh, let's take a look at the cucumber.
Finally.
It's too bad Derek's not here.
Let's zoom in, please.
We found one.
We did.
God.
They look so funky.
We should tell Derek.
Amanda, do you have a favorite sea cucumber?
I love them all. There are these sea cucumbers back in Guam that are like multiple feet in length, and some people kind of call them sea snakes and kind of get afraid of them when they bump into them in the water. I love them. It's really cool to see how many shapes and sizes they come in.
What's the one that spits out its intestines and it sticks to you?
I'm not too sure what they're called, but
You're familiar with them?
I am familiar with them. Like when I would go to the beach as a kid, the other kids would play with them and throw the sea cucumbers at each other.
Yeah, those are what kids do.
They were like little water balloons.
Oh, there's a little sea pen in the back here too. Probably, oh, I can't remember the name of this. They changed the name of all the rock pens.
Where would you?
Okay, let's keep going.
Line, please.
So we got a question from one of our listeners. If you were a James Bond villain, where in the deep sea would you build your evil lair? And what kind of evil lair would it be?
Ooh, that's a new one.
Yeah, I think it's pretty fun. I just want a room with a view with a whole bunch of snacks.
That's the way to go.
I think sponge dungeon, because you're kind of untouchable in there, but...
So would you be the shrimp in that case?
Yes. I would just enact my evil remotely.
So in that case, that would be the sponge condo instead of dungeon.
It could still be a dungeon.
It could be a dungeon. That's a good evil lair.
Yeah.
Call it a dungeon.
It's also made out of glass, which makes it much harder to break without hurting yourself. Right? It's a glass sponge.
Menzel, do you have any evil lair ideas?
I don't know. I have to make it some kind of camouflage where no one can see me. Maybe a volcano.
The bottom of the Palau Trench.
Oh, wow.
Nobody can reach you down there.
You'd be surrounded by the batfish and the ghost sharks.
Maybe I'll find the lost city of Maduriptal. That's the breadfruit tree that I was talking about.
Oh, cool.
Yeah.
What's the name of the city?
Maduriptal. That's the name of the breadfruit tree is Madhu, and then Riptal is the name of the Ngiptal is the name of the village. So Maduriptal is the name of the whole thing. Yeah.
Amazing, thanks.
Okay, hang on 1 second on this coral if you can.
Okay. You can hold position.
Nope. Okay. Sorry, we can move on.
Okay.
If you zoom out and pan right a little bit, there may be. I'm trying. There is another coral. No, it's behind us now. We'll keep going. We'll see another one.
Okay.
Okay, it might be this one to the right of us, actually, that we want to take a look at. That right there.
That one there?
Yeah.
Okay. Let's zoom in, please. Doesn't look too happy.
Yeah, all the polyps are closed up and drooping. Do we have any more zoom?
We do not.
Okay, I can help with that a little bit. Just try not to disturb this bamboo curl that I'm hugging with the craft right now.
Okay. Well, let's move on.
Okay.
Just for reference, Josh, my heading right now is 090.
Yep.
It almost looks like the same rock formation in the northern waterfalls of Palau. It's crazy.
I think it's a similar kind of rock.
Oh, wow. Let's look at this.
Yeah, it's also volcanic rock in the waterfalls of Palau.
Right, yeah.
So
Go zoom in, please.
Okay, this might be...
Plumarella.
Yeah. Great, thanks.
Okay, we'll come wide, please.
No, more like Plumarella.
Apparently so. I am looking through the guide right now, and these things are very difficult to distinguish. we have a second while the ship catches up, we can zoom on that one.
Okay, I'll zoom in, please.
Another primnoid. Not Narella, maybe?
So Christopher is saying...
might be Narella, Narella dichotoma. Okay. Okay, let's keep going.
Slide, please.
Big anemone, that's Venus flytrap anemone, yeah. Okay.
So to our viewers just tuning in, we are currently exploring an unnamed seamount west of Bubble Daob Island, outside of the Palau National Marine Sanctuary. This is around a 16-hour dive, and this dive track was chosen because it has a variety of different habitats and depth ranges, and we're here to understand the presence and distribution of different marine habitats with high conservation value. If you have any questions, comments, please send them over on nautiluslive.org.
Go zoom in, please.
This is Swiftia, maybe? Yeah, okay, it's got the orange polyps. Can we look at the large white one?
Squat lobster?
Yeah. Squat lobster, brittle stars, crinoids. Lots of animals associated with this coral.
It's really creating a habitat for many different organisms.
Yep, there's a little hermit crab and a shell at the top there.
Wow, they're living the life.
Yeah, and this looks like Plumarella, this particular coral.
So do the brittle stars harm the coral that they're settling on?
No, not that we are aware of.
There's some interest in a collection of the Plumarella.
Oh, there is. Okay. Sorry, guys.
Yep. And then we'll have to try and back up like.
Let me make sure we haven't already collected something similar.
So we measure from Atalanta to wherever you want to go, and that's kind of like, you know what I mean? Yeah. Or actually, sorry, you're right. Measure from the back of the ship. Yeah. Yeah, start with 35.
So Christopher Kelly is saying that the crinoids should belong to the genus Glyptometra.
Not yet. Can always come down if we need a little extra.
The crinoid?
Yep. What's the distance apart of them right now?
So one of our viewers commented that there's a bit of a tapping noise that keeps going off. Do we have any idea what could be causing that?
Yeah, we're all right, I think.
Yeah, I hear it too. It's like something's bumping into one of the mics somewhere. Seems to be gone.
Cool, thank you.
So Josh, when you get settled, there is some interest in trying to collect one of these pieces that has a crinoid on it as well.
Okay. See if I can settle in for a photo here first.
Okay.
Might end up being a pretty big piece if I end up having to go, because they're kind of like right in the middle of the coral.
Yeah, I'm not sure. After we get these, well, maybe right now we can just kind of tilt up a little bit and zoom in and see.
Shot here. Okay, we'll zoom in, please.
Yeah, if we zoom out, now that we've got that position fixed, so that actually is at the base of the dead material, so it's not going to get collected with a lot of live material. The one on the left might be
a
better choice.
Okay, I'll have to come around. Are we good on the stills?
It still looks though, well, I mean, hold on here a second before you move the vehicle, Josh, because I'm looking left and you're right. I mean, we'd have to take quite a large chunk of the coral to get the crinoid on the left as well, wouldn't we?
Yeah.
Yeah,
I think pretty tall.
Yeah, I think maybe we should not do that, and we'll just take a
similar to the other one, just a little snip off the side.
Yeah, I'll just take a little snip off the side.
Okay. Okay. Let's come wide. Do we know where we're going to want to put this one?
I was just looking at that. All of our starboard bio boxes have something in it.
Okay.
So we just would need to decide. I think we'd probably be safe putting it with the rock sample.
Yeah, I think that'd probably be okay.
All of our slurp containers are available.
Okay.
And we have a potential mobile organism in the forward bio box, so I don't want to open those.
Okay, right.
Right.
Yeah. But maybe that large one in the back only has a single rock in it.
Yeah, we could put it in there for sure.
Okay.
Another cutthroat.
Yeah, all the rest are other coral snips, so I'd like to avoid that until we have to.
Okay. Are we good with the stills shots? Can I slide over
yes,
a little bit?
We're good.
Okay. Okay, zoom in, please. That's good there. And a little more zoom, please. That's good. Okay, we'll come wide, please.
Great.
Let's come wide.
Sorry about that.
Yeah,
I'm trying to diagnose the little audio popping.
Oh, okay, yeah, no worries.
Correct, 009.
Yeah, I wonder if there's a microphone on somewhere else in the ship, maybe.
So we're just going to go for that, the big one on the back.
Ready for the box open?
Yep. Should be good there.
Okay.
Okay. Close the box. Okay.
Okay. Thanks.
No problem.
Max, we could try muting mics one by one. I don't know.
You could, but I just went through all the cameras and looked around and I don't think it's any of us. I think it might be an external cam or external mic. Just give me one moment. I have a theory.
Okay, are we carrying on to
Yes, we are.
waypoint 3?
Yep.
Okay.
is wrong. Sort of sounds like someone using a mouse. Maybe it's like the data lab has their mic on or something. I have no idea.
I swear I don't hear it at all. I hear no extra sound.
Do you hear it?
No. I hear it.
Yeah, it's almost like a tapping or something.
Oh, really?
What do we have here?
Yeah. Let's zoom in on that, please. Oh, I see. My brain was not.
Yeah. We've got a paramurcied or a swifty or something next to big sponge, glass sponge. Pleura is the sponge. We've seen a few of these on the seamounts that we've visited thus far. Can we get a little bit more of a zoom just really quickly on the coral, and then we'll move on.
Okay. There's some more. A snail there too.
Oh yeah. Very cool little snail. Great, thank you.
Okay, we'll come by, please.
Hemicorallium is the red coral.
Got a fish in the upper, straight ahead.
Oh, yeah. Rat tail up and to the left. There it is.
Not one that's photo taken.
Can we get a partial zoom?
Yeah, we'll zoom it, please.
So why are they called rat tails?
It looks like, yeah, that looks like what we were seeing before, what we're calling Kumba. Yeah. So the question was, why are they called rat tails? And I think it's because some early scientist who was first describing these and seeing their long, kind of tapering, scaly tails thought they looked like a rat's tail.
There you go, come by, please.
Okay, let's keep going.
Zoom in, please.
What is inside that, dead sponge?
I wasn't sure if it was an anemone, but now I'm changing my mind. I don't know. Is it?
It's like an anemone.
Is it? Okay.
Yeah, it does look like an anemone.
Bridge, can we stop the ship here?
And are these ones outside the hydrides or what?
I think we'll be quick,
they could be zoanthids on the outside. Oh wow, how interesting that they would grow on the inside of the sponge, because all of those old spicules, the brown stuff, is blocking their tentacles from intercepting food. It's really interesting.
Are there, is there anything eating the anemones?
There probably are invertebrates that will eat them.
Pycnogonids do, right?
Yeah, pycnogonids suck the juices out of the side of anemones.
Does that kill the
anemone? Most
anemone?
fishes don't bother eating them. Stinging cells and watery. Sea stars will eat them. Yeah.
Maybe they found a good hiding spot in there.
Well, that's a solid, that's some good arms on that squat lobster. Let's zoom in, please. Wow.
Oh.
That's impressive.
Yeah. Like how many times the length of its body?
Just ready.
He kind of skipped leg day though.
Can we can we turn the lasers off for a second?
Sure. Can you get those right?
Yeah, I got them. There you go.
And wow, we're going to want to look at the little polyps there.
Okay.
As well.
Let me settle down here.
So why are they called squat lobsters?
I'm going to sit for a second, Mac.
All right. You want me to go wide?
Yeah, sure. It's going to bump pretty hard here for a second.
They're good squatters.
Wow. Striking a pose.
Yeah.
It's their, I think it's because the abdomen, it's folded back onto their belly.
Okay, so
whereas the other kind of lobsters, the ones we are used to, they're long, right? Their abdomen is like extended outside of their belly.
So like they're doing a squat.
Yeah, exactly, exactly.
Cool.
This coral also seems kind of weird.
It is. Can we pan or tilt up a little bit? Because there's, in addition to these sort of regular branches, we've got a couple of these really long branches sticking up all by themselves. Can we zoom out just a little bit? That's very bizarre.
Almost like they're growing separately from the others.
Yeah.
Have you ever seen anything like this, Menzel?
Yeah, but it's usually uniform and not like these two other arms are way too long.
Is it possibly more than one animal?
It looks like it's all connected. That's not a bad question, but it looks connected. Can we zoom out just a little bit more and tilt down?
It's just copying the squat lobster.
Yeah.
They go to the gym together.
Yeah. One of our viewers said, Larry the lobster is getting his lift on. Don't judge him. And someone said it's the Charles Atlas of squat lobsters. So cool.
Okay, can we zoom on the, it is very cool. Let's zoom on these little yellow polyps down below this black coral.
Okay. Yeah, go ahead. Zoom in.
There's something funny about a lobster being known for squatting that skips leg day.
This looks like maybe a Acanthogorgia or something down here. Okay, we can zoom out.
Okay.
I'll get a couple good stills of this guy.
I did.
Greta? Awesome.
Hopefully in focus. But yes.
All right, let's move on.
Okay.
I imagine there's an interesting evolutionary arms race with squat lobsters where the more muscular claws they have, the better at defending themselves they are, but also the more delicious they are, right? Or the more meat they have. I wonder how that works out for them.
If you could eat a squat lobster, how would you prepare it?
I think people do eat squat lobsters.
Yeah. But how would you prepare it?
I have no idea. I'm a vegetarian, so.
Oh, cool.
Maybe a question for Jeff.
Yeah, the expert in eating weird things.
Yeah, I think we can.
Well,
keep moving.
We used to catch a different family of squat lobsters where I grew up.
Bridge
in Washington
nav.
State. And
We go another 60 meters on bearing 132.
And we would just boil them right along with the shrimp. And they just had a little less meat in the tail.
Did they taste better than the rat tails?
They tasted way better than the rat tails.
Don't give me ideas.
What's that? Well, feels like they're backwards. Yeah, right? Yeah. Anyway, sorry. Little engineering break there.
These two arms on this black coral kind of look like those crazy feathers that come off of the tail of a bird of paradise or something like that.
Yeah, that is
so bizarre.
Very unique. I don't know that I've ever seen anything like that.
I think this black coral must have been damaged or something, which caused it to grow in a strange fashion.
You're sure it's on a mating display?
Yeah.
So there's the background settings to those valves that you don't have access to, right? Like there's a baseline that's set in the background. So if those numbers are different, then that's why those ones you can control might be something slightly different. So like, yeah, we have 100% to play with, but that might be 40% of the range of the valve. Got some catching up to do here. Sorry. Behind you. Sorry, I gotta zoom up a little ahead here, folks.
You can't say zoom unless video's gonna.
Oh, sorry. I have to move.
Bad choice of words.
You have to spell it out.
Direction to catch up.
For a dog.
I see these brittle stars within some of these glass sponges.
Mm-hmm.
It seems like they are trapped as the urchin, the shrimp.
The shrimp? Yeah. Some of them could be.
Can I ask that everyone just like make sure their headset is plugged all the way in? I'm not sure that would cause a problem. Just
it sounds to me like somebody's got a open mic somewhere sitting down on a table or something like that.
That's what I think too, but
you think the lounge is
the lounge
on or something?
Typically doesn't have a headset, but yeah.
Oh yeah, that's true.
And they have to force talk to us.
Yeah,
okay. Right.
Possibly in the data lab.
Yeah, that's
but
what
that's
I was thinking.
Also not a headset usually, it's a mic thing, yeah.
Yeah. But if the microphone is on, you'd be picking up.
The microphone was on, yeah.
Mouse clicking or something.
Mouse. Somebody processing multi-beam data. Click, 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 click. Ooh, what do we got here?
Yeah, let's zoom on that really quickly.
Zoom in, please. Oops, lost it there for a sec.
Do we have any more zoom to really look in at the polyps?
We do have a little bit more zoom.
I'll get a little closer and then I'll come down.
Hey.
Okay, best guess, Stauropathes. Okay.
Okay.
Let's keep moving on.
Yep. Come wide on the video, please.
Okay.
Just gotta catch up a little bit.
Yes.
Urchin.
Oh, there's a cool urchin right there. Let's take a look at that. We have not seen many of them. That looks like might be Sparosoma or something like that.
Let's do a real quick look and then I gotta zoom ahead here. Go, zoom in, please.
Yeah, probably at least in that group. Great, okay.
Okay, come wide, please. I'm just gonna move ahead here so I can pass off to Jake.
Yeah.
Okay, we'll come wide.
Hello.
Audio check, 1, 2, 3.
Want to look at that Walteria while we wait?
Oh, is that?
The sponge.
Oh, you want to look at that? Yeah.
Yeah, let's look at it.
Volume is way down. Josh has this fancy mic.
Oh, fancy.
Fancy mic.
It's got a lot of brittle stars on it.
And push in there.
This is Walteria flemmingi. It's a glass sponge. In the family Euplectellidae.
Whoa.
Yeah. It's got a lot of associates. Lots of brittle stars, lots of amphipods.
It looks like gridded.
It is, yeah.
Very
so the spicules are coming together in this really fun lattice.
Perfect right angles.
That's how you identify it to that species.
Oh.
The other Walterias don't have that lattice work like that.
Wow. All right, come my turn.
Do you want to get moving?
All right.
How's the back row doing?
Yeah, we're just getting set up back here.
Okay. Yeah, let us know when you're.
So yeah, I think we'd like to get moving. We're gonna head sort of towards waypoint 3 right now, and keep our eyes out for any fish along the way. We've collected nice rock and several corals, and we're looking to make a little bit of headway. So we can go ahead and make a ship move towards waypoint 3.
Okay. That's going to be 140.
Thank
140. Roger.
Right. Bridge nav. Good afternoon. We would like to make a move, 20 meters, 140. Yep, thanks. Want to push in and see what's on that coral?
Sure. Push in there.
There's a little crinoid and shrimp. This is a Narella, a type of primnoid coral.
Nice.
Fuzzy?
Very fuzzy.
So how we tell it's Narella, or primnoid in general, is that the polyps have these little scales along the body. Narellas have 3 tiers of scales, and the polyps will close downwards towards the center axis of the branches.
Awesome. I think we're good.
Okay. You can come wide, Aaron.
And that little anemone is a tube anemone, or a Syrianthirid.
Very cute.
So these are a different type of primnoid that we're seeing here. These are Calyptrophora wyvili. They have a unique branching pattern, and they almost always have this snake star kind of acrobatically positioned right in the middle.
And do they always have one?
Push in for a snap zoom there.
One snake star?
Yeah.
Yeah, it's usually just one, and the branches, the way it branches at the bottom, it'll branch out to the sides and then out like 90 degrees from each other to create this sort of basket, you see? And then the snake star tends to suspend itself right there in the middle. May, have we collected a rock today?
We have collected a rock, so we'll be all set on that until we get a little bit more upslope.
Sounds good.
Yep. So maybe we can do some introductions. What do you think?
All right. Well, good afternoon, everyone. If you're tuning in to the live stream, we have just done a shift change, so we're gonna do a little introduction. So we're gonna start with me. My name is Blonak Kichocho. I am from Palau. I am the Science Communication Fellow, and I'm here to observe and kind of see what's going on and try to bridge some things around. Ooh, a shrimp. Yeah, and maybe we could just go towards my right.
Yeah. Hello, welcome everyone. My name is May Lubetkin, and I am the watch lead on this watch and the science manager on the ship.
Okay, Ali everyone, and good afternoon, wherever you are in the world. My name is Else, and I am a scientist on the 12-4 watch, and excited to see what we'll see.
Hi, I'm Taya. I'm just a guest trying to help out on the 12 to 4 shift, and I am the data logger. So.
In your front row, sitting in the nav seat, this is Megan Putz.
Megan, that was an incredible intro. Your voice was so, like, radio smooth.
I'm trying to do the Dan. Dan voice.
Good luck following that.
And my name's Jake. I'm driving the bus. I am the Herc pilot in this watch.
I am Sarah Sargent. I am the Atalanta pilot.
And I'm Erin Rainey, and I'm your video engineer this watch.
All right.
So Megan, approximately how deep are we right now?
We are 1267 meters.
1267 meters. Got it.
That's the
Venus flytrap?
Yeah. Oh yeah, it's a Venus flytrap anemone, or it's an anemone in the family Hormathiidae called Pheliactis. There are two anemones that get the name Venus flytrap, and this is one of them. The way to tell a difference is that these ones often perch on rocks and have sort of a brown, kind of rough, bumpy skin, whereas the other Venus flytrap tends to be pink and smooth and will attach to corals or sponges or anything up off the bottom. And that one's called Actinoscyphia.
Are they found in the same regions then?
Yes, you can see them right next to each other oftentimes.
Oh, cool.
But they both have this sort of folded morphology to them that give them that Venus flytrap-esque look. But they are not related to each other. We'll probably see an Actinoscyphia later. Usually we'll only see them when we see lots of coral.
How large can those, like, Venus flytrap anemones get?
That Pheliactis, that's usually about the average size, that big kind of, about 30 centimeters.
Every time I hear, like, the Venus flytrap, for some reason, I think of the Little Shop of Horrors movie.
Yeah.
Maybe we'll see a giant one.
I saw some really giant anemones the other day.
You did?
Yeah.
When you were diving?
When we were diving.
Oh.
It was so amazing.
At the beginning of this dive, that was one of the first things we saw was a really big anemone this morning.
Yeah. Was it one of the actinostolids that have those, like, bulbs on the ends of the tentacles?
I'm not sure about that. It looked a little bit more like a Venus flytrap, but...
Okay.
Bamboo coral?
Yeah, let's zoom on this bamboo.
Yeah.
All right, push it.
So high density of polyps on this bamboo coral. But you can see those black bands. Those are the nodes of the coral where the coral can articulate. So it has two pieces to its skeleton. This sort of hard calcium carbonate sections that act kind of like bones, and then these flexible proteinaceous sections, those are the black nodes that allow it to be very flexible, giving this whip the ability to move freely in the water. We do love a good whip coral. And can we look at the base?
Sure.
Yeah.
Come wide a bit.
Yeah. sometimes looking at the base and seeing where that first node starts can help identify it. These bamboo corals have been going through a lot of revision lately. We've been learning a lot more about how to identify them, doing a lot of genetic testing on samples that have been collected, understanding a little bit more about the diversity in this group. Thanks, Jake. That's great.
Yep. All right, come on.
Bridge, Nav. Can we make a 30-meter move 140? Thank you.
140.
I guess we got channel 2 back on. For our viewers who are just tuning in, we have channel 1 and channel 2, but channel 3 is still being troubleshooted, so it should be up shortly.
For those of you who are just joining us or who have been with us since the beginning of the dive, we are making our way upslope on an unnamed seamount that we mapped just this morning, shortly before we dove. And we're trying to get up to the summit of this seamount, which is around 690 meters. We're currently around 1,200 meters, 1,268 meters to be exact. And the primary goal is to characterize the various kinds of life and biodiversity that we're seeing along the seamount at different depths of different species, the abundance, as well as the geologic setting by collecting some rocks. And another big primary goal of this dive and of this cruise is to collect eDNA water samples, which is a goal made with our partners at Pickrick. Elsa, do you want to share a little bit about the eDNA water sampling?
Sure, thanks, May. So Pickrick, which is the Palau International Coral Reef Center, is